Welcome to the problem-solving set on genes and the environment. Question 1. In a breed of roses, thorny stems are determined by the dominant allele, big T, while thornless stems are determined by the recessive allele, little t. The dominant allele is 90% penetrant in the heterozygous plants. In a monohybrid cross, what are the expected ratio of phenotypes observed in a population of 1,000 offspring plants? So before we take into consideration penetrance, let's determine the expected number of offspring. Now this is a monohybrid cross, big T times big T little t. So for this, you would expect a quarter to be big T, a half to be big T, and another quarter to be little t. Now they're saying this is a population of 1,000 offspring. So a quarter of 1,000 is 250. One half is 500, and the other quarter is 250. Now these are the expected numbers. What the problem is saying, however, is that the dominant allele, big T, is only 90% penetrant in the heterozygotes, which means that only 90% of the heterozygotes, although they should all have thorns, only 90% actually do. So now let's look at what the actual numbers are. For the homozygous dominant, big T, it doesn't say anything about an altered penetrance, so we'll just say that 250, when they have both dominant alleles, they do have thorns. For big T, little t, only 90% actually have the phenotype expected. So we'll do 500, which is what we were expecting, times 90%, and that gives 450 that actually have the thorn phenotype. Now that means that the other 50 do not have thorns, or are thornless, so we'll add them to the thornless little t, and that will give 300. So the total phenotypes, these two, which total 700, have thorns, and these 300 are thornless. Question 2. Dimples are generally inherited in a dominant fashion. Assuming this characteristic is 80% penetrant, what is the chance that a child born to a heterozygous father with dimples and a mother without dimples will have this trait? So let's do the genotypes of the parents. The father is heterozygous and has dimples, where the mother doesn't have dimples. Now if you cross them, half of the offspring are expected to have dimples and be heterozygous, and the other half are expected to be homozygous recessive and do not have dimples. So we would guess that it would be one half of the children. However, this characteristic is only 80% penetrant. So combining those two, you have the half chance of being heterozygous for dimples, but then only there's an 80% chance of actually showing this characteristic. So that equals a total of 40% chance that a child will have dimples. Question 3. Some genetic students get confused with penetrance and expressivity. Contrast the two terms and give an example of each. So penetrance. This term means the percentage of those with a genotype that actually express the expected phenotype. So say those with genotype expressing and this is usually given as a percentage. Now an example of this would be, as the previous problem, someone with dimples, the dimple dominant allele, but not actually having dimples. So, or if someone had the dominant allele for freckles, but then had clear skin. In contrast, expressivity is actually the extent of a phenotype in a given individual. And an example of this would be if somebody had brown eyes, they had the genotype big B, and they had light brown eyes, whereas their brother had big B, and had dark brown eyes. Or another example is if a female has a cystic fibrosis allele and she has respiratory problems, whereas a male with cystic fibrosis is infertile, and both of those symptoms are due to the disease, but they have different extent in the phenotype of these individuals. Problem 4. Overdominance can be described as heterozygous advantage. An example in humans revolves around the sickle cell allele. Carriers of this allele have normal red blood cells but have an advantage of being resistant to malaria. Individuals homozygous for the sickle cell allele have blood disorder called anemia that can lead to a lower lifespan. In populations where malaria is rampant, this allele is overly selected for. In a cross between a malaria-resistant carrier and an anemic individual, what is the expected phenotypic ratio in the offspring? So let's define our alleles. Because it's a sickle cell allele, we'll use the symbol S. So we can say SS is normal. They don't carry the sickle cell allele. S prime S, and these can be resistant to malaria, but they have the normal red blood cells. And S prime is anemic. Okay, so for A, we have a malaria-resistant carrier, S prime S, crossed with an anemic individual, S prime. And what are the phenotypic ratio in the offspring? Well, this individual can only give the S prime allele, and this individual can either give the S prime or the normal S allele. So the offspring would be 1/2 S prime, which is anemic, and the other half of the offspring would be S prime, which would be resistant to malaria, but have normal red blood cells. For B, what is the expected phenotypic ratio for the offspring of a normal individual crossed with a carrier? So now, this is a normal individual crossed with a carrier of the sickle cell allele. This individual can only give the normal allele, and this individual can either give the sickle cell allele or the normal allele. So in this cross, it would be 1/2 normal and 1/2 S prime, which would be malaria resistant with normal blood cells. And this is resistant, sorry.